I study Arctic grayling on the north slope of the Brooks mountain range, where climate change is happening very rapidly. Studying the grayling in the Arctic can give us a handle on issues about habitat fragmentation and population resistance to climate change and resilience to climate change.
Once you know how far a fish moves and when it moves it, that gives you a whole context on what else the fish may be doing. Once we apply some radio tags and some other techniques, we've realized just how extensive some of these migrations can be.
And when the fish goes through that antenna, bloop, we know we've got its number, we know where it was, and we know what time it passed through the antenna. We've been using PIT tags, which are passive integrative transponder tags. It looks about the size of a Tylenol, and you inject that into the animals. It's kind of like Easy Pass. If you have Easy Pass on your vehicles and you go through the Easy Pass, bloop, it knows you've been there, it knows where you are, and it sends you a bill. With the fish antenna arrays, we have the antennas set up in the river, all along the river.
This river freezes essentially solid in the wintertime. So the fish could not stay, and in fact we've documented that they don't stay. They migrate in mid to late August. They migrate upstream about 20 to 25 kilometers upstream into a headwater lake where they overwinter. So if you were to come here and fish in late August, you would conclude there were no fish.
For the Kuparic River grayling, Green Cabin Lake is their overwintering lake. It's their major overwintering lake. It holds, we think, probably about 5,000 grayling.
When we started our work in Kuparic, one of our thoughts was to determine just how far they migrate. A lot of people think fish don't swim very far. They spend their entire lives, say, right outside your village, right out close to where someone would catch them. But once we apply some radio tags and some other techniques, we've realized just how extensive some of these migrations can be. I'd heard of Linda Deegan's work, so I knew that there was a considerable database of historic information on grayling in the Kuparic. We started putting our heads together thinking about how would we describe the potential effects of climate change, how that might affect the grayling population. In the context of grayling being an indicator species of other drying trends that could occur on the north slope with other species. And so in 2010 we implanted radio tags, radio transmitters, into 100 Arctic grayling. Because of Linda's work and Heidi's work and Cam McKenzie's work and such, we knew that the place to do this was at Green Cabin Lake where they overwintered. The concept was the whole population overwinters there, so it would provide us easier access to actually capture these fish and surgically implant the radio tags. We had to implant the tags in the fall and let the fish overwinter with the tag inside of them so that we could follow their movements the following spring.
The PIT tags are always active because it's actually the antenna that's doing the reading. It's different from the work that Jeff is doing with the radio transmitter tags. Those have a battery in them, and the tag itself is doing the transmitting, whereas the PIT tag doesn't have any battery. It's just a coded wire tag, and when it goes through the antenna, it's the antenna that's picking up the tag.
The interesting thing is the grayling and the Kuparic know to migrate upstream. There are fish in other rivers and streams that will go downstream to springs that have flowing water all winter long.
From this data, we found that their movements are highly tied to the hydrology of the system. So in the spring, they move out, they spawn, but in the fall, their movement back into the headwater lake is really tied toward, first, we think probably some trigger like photoperiod, and then river hydrology. So when it rains, those flood events trigger movement and they move up to the lake. When the connectivity of the river is impeded by a drought, which happened in 2011, the fish movement patterns were just totally altered. So they could not make it up to the headwater lake. The fish got piled up at a dry zone and they were greatly delayed in getting back to the lake. And we were able to catch a bunch of fish caught in the pool and monitor their weight loss over time, and they actually lost like 30 grams of weight while stuck in the pool on average. That's a huge amount of weight, especially if you require that to then be able to overwinter and spawn the next year. So you can't meet those two thresholds, survival and reproduction, if you don't have the resources available to do that. Getting caught in the river delays the migration back to the lake and we think increases susceptibility to mortality. Probably not a great thing for the population if that type of thing happens over and over. As a child, I was always in the tidal pools. I grew up on the shores of Connecticut, and that's where I spent my summers, is like looking into the tidal pools and trying to figure out where the animals were moving to and from and why. And I even once tracked my cat out the bedroom window on the 3rd floor to see how she was getting to my bedroom at night, you know. So I've always been curious about movement patterns and why do animals move where they do, and nature in general. So, you know, that is sort of my passion and driving force, is why and how.
Grayling are very common. They probably don't receive the love or the notoriety that they should.
Arctic grayling is in the family Salmonidae, which means that it's related to salmon and trout and whitefish, all the salmonid species.
There's a ton of other species, at least on the recreational side, that are usually much more popular than grayling.
If you're lucky and it's a sunny day, they might leap out of the water at a fly, just give you a nice fun fight on the line. But they're also important for native peoples that feed them to their dogs, and people do eat them.
In certain locations like the Chena River and Fairbanks, they are very, very popular species for rod and reel anglers. As well as in the smaller communities, they're very important seasonally for subsistence fishing. They're usually one of the first species folks capture, and to have fresh fish after a winter of eating dried food and grocery store food, and it can be a real treat for folks.
Some of my research is looking at how variable is the gene pool, how many populations there are across the landscape, what is the connectivity between those, so what's the migration rates between the different populations, but then also looking within a population, what is the ability of each individual population to maintain itself and its birth and death rates through the connectivity of the environment between habitats.
They're very important to the biodiversity of systems. If you look at the distribution of them in North America, I mean, they used to occur in the upper Midwest. They no longer occur there. They're on the endangered species list in Montana. And I think those are different species, but the point being the habitats that grayling need are very cold water, very clean water. And so the places that they still exist in North America are the more remote areas.
I've looked across at 16 different locations near Tulick Field Station on the North Slope.
That's nice.
From those samples, I was able to distinguish distinct genetic populations. So we call them genetic clusters, which means that those different locations are exchanging genes with each other more frequently than they are with the other populations. There's still a little bit of movement between populations, but mostly they're exchanging within those genetic clusters. And those genetic clusters are probably due to watershed boundaries, and those watershed boundaries occurred probably about 10,000 years ago, when those glaciers retreated and left those watersheds as they are.
The Park River from Green Cabin Lake down to Simpson Lagoon on the Beaufort Sea is about 180 miles long or so. And again, it comes out of the foothills of the Brooks Range and then goes through the coastal plain. The different habitats may actually be what's distinguishing that lower and upper river population, if they do exist, because the habitats are different.
If you had one huge, big, like we call it panmictic population, which means that everybody's exchanging and it's just really one huge population, if you think about genetics, that would be just one homogeneous genetic population, right? When you have isolated individual populations, you have the opposite scenario where there's no movement between the populations, and you get these isolated populations where you get a lot of local adaptation going on. So the genes in this population would be very different from the genes over here, but they never interchange. If you get a local extinction over here, you then don't have that exchange of genes. That population is lost and you get a decline in your genetic variation. But if you have some scenario in the middle where you have some isolation and some interchange of genes, that's the best scenario actually for resilience in terms of genetic variability, because you get some local adaptation, which causes changes in the gene populations, but then you get some exchange too, so they share them, and then you get a lot of variability across the landscape.
The next piece would be to try to identify the movement patterns of that lower cupark population within the cupark oil field, see where those fish move, and then at least you'd have a much better definition as to when and where those two populations intermingle, if you would. They may not spawn together, but at least their range may overlap. If that's the case, perhaps the stream is big enough, and again, this is one of those pieces that nobody's done enough work on this critter or most other critters in the Arctic, but perhaps there's even a third location. Maybe there's a discrete group in the middle that lives its entire life more in the middle part of the cupark. So you'd have a lower population, a middle population, and upper, but we don't know that yet. But that would be my next interest, if you try to define what the lower population does.
We can't change the course of climate change. That's here. But what we can do from a management perspective is when we build roads, for example, they're planning a road out to Umeå, which is very close to my study area. We can ensure by knowing what these populations are across the landscape, for example, and what interpopulation needs might be and movement patterns from both my work and Linda Deegan's work and Jeff's work. We can then say we need to ensure that there are culverts placed in these locations and that they're proper, and that we have a bridge here in this location, and just to ensure that the habitats that are critical for these fish to move to and from remain accessible.
I'm Kate Mitchmerheisen, and I'm a field assistant to Heidi Golden up in the Arctic. We would head out to one of the rivers in the morning and start to head up or down the stream, collecting samples from distinct groups of fish. So we had a goal of about 30 from every river reach. You're only doing one reach of one river one day usually, and then you may be at another reach of the same river, but further downstream or upstream. And we'd collect those fish every day, and that's what we did.
Oh, I see it. Okay.
So young of year is yoi. We call them yoi. We're looking to see whether there are more than one spawning population within what we're determining as a genetic cluster.
Do you see it? Up right above a rock.
I found 5 genetic clusters, but if you look at the system more closely, perhaps there's more structure there than you see on the landscape level. So what I suspect is that the different locations for spawning might be different populations that are spawning.
Well, there's 2. There were like 5. Well, now I only see 1. I'll come around this way. Maybe it'll scare it into you a little bit.
To test that, what I'm doing is going out to different locations along the river and then collecting young of the year that have hatched out at that site. Fish tend to school together, so you can actually very easily get a scoopful of 30 fish. But those are not good for genetics because it could be one family that you're scooping up, and then you would bias your genetics.
Do you see them?
The way we're sampling is to go to one location like that where there's a lot of fish, but only take one from that location. And then we move downstream to a totally separate location away from that one and get one fish from that location. So yes, it's very difficult to get the numbers that we need to do the genetic work because of the way the sampling is set up to avoid the bias of getting siblings in your sample. Just because adult fish are together doesn't mean that they're sharing genes. It doesn't mean that the adults are spawning together. They might be from multiple populations that are just feeding in the same location and overwintering in the same location, but not necessarily spawning at the same locations.
Yeah! You got it! Sweet!
Looking at those young at the year, if there's no genetic differentiation between these locations with the young at the year, then we know that this watershed is just one big spawning population.
More downstream? Oh, cool. This is a big fish winner today.
If climate change is causing these dry zones to occur, they might block off the more risky venturing fish. So the ones that go far have a greater likelihood of getting trapped by a dry zone coming back to overwinter. If those fish are weeded out of the population by natural selection, what you end up with is fish that tend not to migrate so much and more isolation of populations. That then has implications for the whole metapopulation, because the fish that are going to exchange genes with other populations and other watersheds are the ones that are going to be the travelers, the ones that are going to travel far to spawn.
What's the total number of yoy? There's 3 in my pocket. 16. Thank you. Nice.
I had this great opportunity because I had been in the Arctic doing research in the early 90s, side by side as a peer with Heidi at that time. And we were very young right out of college. You really absorb as much information as you can about many different projects. I remember hearing talks about thermal karsts in particular, and that it was a rare occurrence. But the theory was that as we experienced warming, if warming occurred, these were just ideas of what might happen, that we would see many more thermal karsts happening in both the lakes and the rivers. And they described the idea, the process of what they thought might happen, and the impacts. And so to go back 23 years later, that it's incredible the accuracy with which they predicted exactly what would happen, because we see many thermal karsts as we go along the rivers. The impact is exactly what they thought it would be. So I hope for people to realize that these predictions, that they're coming from intense observation and an awful lot of data collection. And now, as Heidi says, we take what we have and move forward with
Hey
it.
bears! I wonder if they'll learn. They're calling us. Hey there!
Otoliths are fish ear bones, and the neat thing about an otolith is that it's a little like treasure trove of information. The center of the otolith is the birth of the fish, and as you go toward the edge of the otolith, that's the death of the fish. And it's like a tree ring. So if you look at a cross-section of a tree, you can age the tree. You can tell how much it's growing by looking at the distances between the bands on the tree. And you can even go in and look at the chemical composition of the cells in those bands. You can do the exact same thing with an otolith. So all of that information is contained inside that little ear bone. I've taken 10 fish from each of the locations that I've gone to, and I'm going to be analyzing those ear bones for aging the fish and also looking at the chemical composition, and then match it up to the chemical composition of the water at different locations along the river, and you can actually back calculate where the fish has moved over the lifetime of the fish by matching up the chemistry in the otolith with the chemistry in the river.
And that can tell you about the habitat?
You can see the peaks in the chemistry as it goes from overwintering site, which is one chemical composition, to a spawning location, which has a different chemical composition, to a feeding location, which has then yet another chemical composition.
Little antics there.
Not only that, but you can track it over the lifetime of a fish. So if I was looking at a 15-year-old grayling, I might expect to see not too much variation in the chemistry up until maybe spawning age. Then when the fish reaches spawning age, I would expect to see changes from the overwintering location to a spawning location and then back to an overwintering location throughout the rest of its lifespan. The chemistry is really dictated by the underlying glacial geology of the area. And the North Slope has a very nice, strong signal in its water chemistry because it was glaciated 3 successive times. There was the Itkillik I, Itkillik II, and the Sagavanirktuk glaciations. And the first glaciation extended far north and then receded. And then the next glaciation wasn't quite as far and receded, and the final one was even less far north and receded. So those are three different levels of exposure that the rocks have had. Over time, minerals leach out, the isotopes change over time while those rocks are exposed. So you get these differences. It's a gradient in chemistry from the headwaters down toward the Arctic Ocean. And that gradient is what allows you to use the otolith microchemistry as a signal. If there's no gradient in your water chemistry, then you really can't use otoliths this way. We're really fortunate that we have this great signal in our system. It took me a little while to realize what my calling was, and it wasn't until I took an upper-level ecology course that I realized that all of the things that I was good at, mathematics and biology, I loved curiosity about biology, and also just communicating science and communicating in general, like teaching.
Nice.
Those things are all integrated in ecology. You know, you use mathematics and you study biology, then you use those mathematics to then explain your biology findings, and then you go and communicate it to others. So it was a really good match. For since 1990, when I went with Linda to the Arctic to study trophic dynamics of grayling in the Arctic, I just fell in love with the fish and the Arctic. Even though I'm studying the same species, the questions have changed. So we were looking at trophic interactions initially, and as time goes on, you learn more, and the questions start to shift, and now all of a sudden it's like, oh, the landscape is changing. How is this landscape change going to affect the populations across the landscape? One possible safety net might be the location of alternate overwintering sites, which we think exist and have gone out on snow machines during the winter to try and find these locations. And we did run into one location where there was a spring and the water had not frozen solid during the winter, and there were actually fish there. So if there are a lot of these locations, then that would suggest that perhaps that could be a safety net in climate change where they can't get to their overwintering site. Perhaps some grayling would survive in these pockets of unfrozen water. To figure out where we were going to go, we looked at satellite imagery and some thermal maps that we got from the Toolik Field Station GIS people. From those maps, we located areas that looked like they could be potential wet spots or seeps on the landscape. And so we went out with those locations in mind and traveled the river and looked to see, is there alpha ice here? Is there open water here? If there's open water, are there fish here? We only found one that had fish, and this was along the Kuparik River and along Oxverkoyuk Creek. But we suspect there might be more that just aren't open, that we can't see. One indication of locations where we think there are springs is these features called alpha ice. And an alpha ice is like a land glacier. And the way that they form is that water comes from a slightly deeper source, and it holds just enough thermal energy, maybe just a degree above freezing. It doesn't have to be much. That thermal energy then travels along the landscape. It hits the -40 air temperature and then freezes. So where we find this characteristic alpha ice, you often find these springs. The spring that we found that had grayling in it, that was the alternate overwintering site on the Kuparik River, has an alpha ice downstream of it. The other thing with that alternate overwintering location that we noticed was that there was high mortality there. So we saw a lot of dead fish under the ice, possible that they just ran out of resources and didn't survive. And there were animal tracks all over. The animals come there. It's like an oasis in a frozen desert, if you can imagine such a thing. And if you're a wolverine, they can just go and, it's like the supermarket, and pick a fish out of the freezer. Aside from just visually looking for alpha ice, we put a remote antenna array on the back of a sled, and actually instead of having the antenna in the river, we dragged the antenna over the river, and we were looking for fish under the ice. Using that antenna array, we actually found 3 tags under the ice, and each of those tags was associated with these seeps that we had pinpointed from the thermal imagery. So that was a huge, like, eureka. Now, we don't know whether those tags were ones that were spawned out by fish, because there is a tag loss rate, and we suspect that it's higher with females than with males because of where we tag the fish. So we tag the fish in the abdomen, and if it's a female, it's possible that the female could then spawn that tag out. But the exciting thing about that is that if it is a spawned out tag, that it could indicate that those locations are actual spawning sites. Or it could be that there's actually a fish under the ice. We didn't know whether it would work or not, and we were pleasantly surprised that it did work.
Everything is something to learn about at our house, and whether it's going outside and hiking, camping, investigating everything you can has always been really important to us. But we like to participate in our ecosystem even in the city. We're in the Neponset Valley watershed. That's a watershed, and we can do so many things for our river to make it a clean place. I feel I can bring all my science background with me when I go to community events, school events. My son just brought a whole group of inner city children to a land trust that's really, really close to the city, to get them outside and enjoying being out in the world. It informs everything we do, really, as a family, but just also as a part of the larger community, not just our little neighborhood.